Hi, I'm Anna Kasdan, and I'm a PhD candidate at Vanderbilt University. And today I'm excited to talk about a project called Identifying a Brain Network for Musical Rhythm, a functional neuroimaging meta-analysis and systematic review. So we know that across multiple methodologies such as EEG, MEG, fMRI studies with clinical populations, that rhythm processing involves a cortico-subcortical network. And these brain networks for rhythm processing have been informed by the timing literature, where we know that beat-based timing involves the basal ganglia and SMA, whereas duration-based timing is thought to rely more on the cerebellum. And evidence from clinical populations suggests that these two timing mechanisms tend to dissociate in the brain. And we were interested in understanding the functional organization of a brain network for musical rhythm. And one way to approach this is through fMRI because of its really good spatial resolution. And thankfully, there are a good number of studies that investigate musical rhythm using fMRI. However, many of these studies have been conducted in small samples, and they vary considerably in terms of their stimuli, task demands, and experimental procedures. We wanted to see what would hold up across the idiosyncrasies of any particular study, so we used a meta-analytic approach to identify a brain network for musical rhythm. The first step in this process was to conduct a literature search in accordance with PRISMA guidelines. So broadly, we were looking for studies that investigated rhythm processing using fMRI in neurologically healthy adults. So this could involve participants either listening to or producing rhythms, and all reported analyses had to meet certain fMRI methods criteria for the meta-analysis software. So for example, they had to be whole brain analyses and not ROI analyses. We specifically wanted to look at studies that investigated some main effective rhythms, so not comparing rhythm to melody processing, for example, or some other general music processing mechanism. I won't go into all of the inclusion and exclusion criteria here, but some important ones that we had were looking at rhythms only in the auditory modality, and also only including studies that investigated neurotypical adults, so not children, or studies with clinical populations. We identified 31 studies for our systematic review. Effectively, this is 30 studies because 2 studies were conducted in the same participant sample with the same stimulus, but slightly different tasks. Our next step was to descriptively characterize different study variables. So as I mentioned, we had 30 studies of musical rhythm in the meta-analysis. And one of the first things that became apparent with these 30 studies was the heterogeneity of the literature. So I attempted to bucket this. And so fMRI studies had very different questions they were aiming to answer, different stimuli they were using to answer these questions. So the first way to divide these studies was to divide them based on whether they were manipulating looking at beat-based versus non-beat-based rhythms, or whether they were manipulating rhythmic complexity, so more versus less complex rhythms. I then separated studies within the beat-based category based on the type of baseline condition. So I noticed there were a group of studies that had some sort of auditory control as a baseline, and then there were another group of studies that had rest or silence as a baseline. And so these categories were not only motivated by the fact that there were differences in the literature between studies and the types of tasks and stimuli they were using, but also motivated by frameworks for beat-based and duration-based timing. So the unit of analysis for descriptive variables is studies, whereas the unit of analysis for the quantitative fMRI meta-analyses is contrast, as many papers had multiple relevant contrasts for these separate buckets, and all the meta-analyses were conducted separately, so there weren't any issues of repeated measures or overlapping participants. Okay, so what types of stimuli are people using in the field? The short answer is many types. So this figures my attempt at some dimensionality reduction of terms. And I'll acknowledge that this is one of the trickiest parts of the project, as I had to make some executive decisions about the terms, as authors would use the same term differently across different studies. So I have some audio clips to sort of ground examples of these terms. The first audio clip is an example of an isochronous rhythm, which I think we're all pretty familiar with, pretty standard. The second audio clip would be considered a syncopated rhythm. It was taken from a study by Kornicheva et al. In 2011. And this is an example of a more complex beat-based rhythm as opposed to the rhythm we just heard, which was a less complex beat-based rhythm. This last clip is a non-beat-based rhythm. It's a scrambled rhythm, and this was actually the auditory control for the syncopated stimulus that we just heard. All right. Other descriptive variables we were interested in were the participant sample. So most studies saw non-musician participants. We also were interested in the use of sparse sampling. So fMRI scanners are not only very noisy, but also very rhythmic. So this could be a potential confound when studying the processing of auditory rhythmic stimuli. And the sparse sampling method involves periods of silence between MRI volume acquisitions so that you can present your auditory stimuli without contamination from the loud and rhythmic noise of the scanner. And interestingly, the majority of studies did not use sparse sampling. Next, we looked at the type of stimulus that was used across studies, with the most common being what we're calling a simple sound sequence. So just a sequence of tones in a woodblock timbre or a piano timbre, for example. And lastly, we looked at the types of tasks that participants completed while they were in the scanner. So most commonly, participants were doing a passive listening task or a tapping task, but there were several different kinds of tasks participants were asked to do in the scanner. And I'll mention that in our fMRI meta-analyses, we pooled across contrast looking at perception and production, as we know that these are very intertwined and also activate similar brain networks. And I'd also like to give a shout out to my color palette, which is an R color palette of the women of the 2021 inauguration, and you can download it at this GitHub link here. All right, so now for our coordinate-based fMRI meta-analyses. Most commonly, I think the method people use for fMRI meta-analyses is the ALE method, but I decided to use a method called seed-based demapping, which assesses the effect size of activation or deactivation for each voxel, while ALE does something a little bit different, where it measures the degree of overlap or convergence in peak activations. So SDM uses standard univariate voxel-wise tests rather than testing if findings tend to converge in a given voxel. All analyses or contrasts had to be whole brain, and for each separate meta-analysis, we chose the best fitting contrast per paper. One important thing I think to mention, not only for fMRI meta-analyses, but for meta-analyses in general, is that they're only as good as what is already out there in the published literature. Okay, so our first fMRI meta-analysis was looking at beat-based compared to non-beat-based musical rhythms, and we found 4 clusters of activation here in the bilateral putamen and in the left precentral gyrus and the left Rolandic operculum. And I'll mention that all maps here are thresholded using threshold-free cluster enhancement. Unsurprisingly, we get more and larger clusters of activation when looking at beat-based rhythms compared to a rest baseline. So we see the bilateral SMA, we see large clusters in the left and right temporal lobes, we see bilateral activation of the cerebellum, as well as larger areas of the basal ganglia that are emerging as clusters. And one thing that was exciting to me here was it seems to be, at least from this meta-analysis, that rhythm is bilaterally represented in the brain. The next main meta-analysis that we did was looking at more versus less complex rhythms. So for example, syncopated compared to isochronous rhythms. And we found several activations here. So we found a cluster in the bilateral SMA, mid-cingulate gyrus region. We found activations in bilateral cerebellum, as well as the left precentral gyrus and also left and right parietal lobules. We also found 2 clusters of activation for the reverse contrast, so less versus more complex rhythms. So this would be looking at rhythms that have a stronger beat structure compared to a less strong beat structure. And these were in the left putamen and left STG, again, implicating the putamen in processing of beat-based rhythms. So to conclude, it seems that rhythm is largely represented bilaterally in the brain, and the choice of a baseline condition is crucial to determining a more specific brain network for musical rhythm. The bilateral putamen emerges as an important area for beat-based musical rhythm, and the SMA cingulate gyrus region, cerebellum, and parts of the inferior parietal lobule seem to be modulated by rhythmic complexity. The field has diverse types of stimuli and tasks, but importantly, findings tend to converge and we get some interesting clusters of activation despite these differences across individual studies. And I think this work has several interesting implications and future directions, one of which is I think it can inform the design and implementation of future fMRI studies of musical rhythm. And it also gives us a basis for understanding how the brain basis of rhythm relates to other cognitive functions such as language. So with that, I would like to thank my meta-analysis team, our funding sources, and I'm excited for questions and discussion. Thanks so much.